I am really delighted to introduce our first keynote speaker, Dr. Ben Langmead. He earned his PhD in computer science from the University of Maryland in 2012 and is currently an associate professor of computer science at Johns Hopkins University. His group really focuses on ways of making biological data sets easy to use for biomedical researchers. And specifically, his group studies and applies ideas from sequencing alignment, text indexing, statistics, and parallel programming. You may recognize the name of several widely used software tools, such as Bowtie and Bowtie 2 from his group. And current work in his group involves methods and software for efficient sequencing data analysis, methods and resources for making it easy to reuse and query archived sequencing data sets, and methods for alleviating bias and characterizing uncertainty in read alignment. He's the recipient of several prestigious awards, including a Sloan Research Fellowship, National Science Foundation Career Award, and the Benjamin Franklin Award for contributing to open access. So he's been a longtime member of the RECOMB-Seq community, so I am really delighted to introduce him. Let's welcome Dr. Ben Langmead.
Thank you. Can you hear me by any chance? Okay, great. All right, I'll start the screen share and you can see this. And perhaps can you also see a red pointer?
Yep, you're all good.
Okay, all good. All right, fantastic. Thank you very, very much. I'm thrilled to be here. Yes, it's true. I love RECOMB-Seq. I'm delighted to be able to come here and give a keynote talk. I feel like in keynote talks, part of the responsibility is to kind of zoom out and do some synthesis of different ideas and give a perspective, some historical perspective and some future perspective. But yes, I will also be spending a little bit of time flogging methods from my own group. So let me get started. Let's start with the human reference genome, something that probably everyone here is pretty familiar with. You've probably loaded it as an input into some bioinformatics tools. Maybe you've built indexes of it, and you've had to call it by one of its many names, like GRCH38 or HG19 or something like this. And you probably know that if you open up the file that contains this reference genome, what you're looking at is a collection of strings, one string per chromosome. Right? You could print it out in a series of books, and it would just be a linear string. You may also know that in the Human Genome Project, the way they sequenced the human genome originally was through this process where they first created a genetic map to kind of split it up into sections, and then they assembled each section separately. But at the end of the day, what they got was one haplotype. It was a haplotype that was basically a stitched together series of sections from different people's genomes, but it was just one haplotype, a single string, right? And so we often just call this the single linear reference genome. And this, this is the reference genome that we've grown up with, and it is good in its own way, but it's got a we've had a consistent complaint this whole time, which is that it's not really designed to be representative of large numbers of people, right? Increasingly over time, we of course have learned a lot more about population genetics, what polymorphisms are out there. The Human Genome, the 1,000 Genomes Project obviously surveyed a great deal of polymorphism among humans, genetic polymorphism among humans, but there's many more examples. There's, for example, the Haplotype Reference Consortium, TopMed, ongoing sequencing efforts that are going to sequence millions of people. And so we're continuing to build larger and larger catalogs of how people actually vary genetically. And then on top of that, we're also conducting lots of new assembly projects where using long read technologies, especially things like PacBio Hi-Fi and Nanopore, we're able to assemble high quality human reference genomes. These are several examples, but I'll highlight the bottom one there, which is the CHM13 T2T assembly that, among other things, is what Adam Philippi spoke about in a keynote a couple of years ago in RECOMB-Seq. So with all of these advances, with all of this new population genetics information, all of these new genome assemblies coming from long reads, it becomes seems increasingly silly that we use a single linear reference genome as the basis for all of our analyses. So that's one way I can justify sort of the topic of this talk, but let me also flip it around and say more sort of specifically and positively, the reason why we need to go beyond the single linear reference sequence is because it causes an actual collection of problems, which we call reference bias, right? We have this kind of bucket term of reference bias for all the things that can go wrong when we are using a single linear reference genome as our point of reference. Let me be more specific here. So specifically, reference bias refers to the fact that when we have polymorphism and we have a reference genome that is just a linear genome, the reads that come from the polymorphic parts of the donor's genome might not contain entirely all reference alleles. It might have some non-reference alleles or alt alleles. And when reads contain alt alleles, that creates mismatches and gaps in the alignment of that read to the reference genome. And that in turn makes it harder for the aligner to get the right answer. Right? The aligner has a reduced ability to find the alignment in the first place because the mismatches and gaps might, for example, disrupt its heuristics, or it might drive the alignment under some score threshold. Or if there's many choices for where the alignment could go, for where the read could go, these mismatches perhaps confuse it into picking the wrong one. So reference bias is our bucket term for the sorts of mistakes that aligners can make when reads contain non-reference alleles. So to be more specific, a kind of bioinformatics analysis that this can confound would be, for example, any allele-specific analysis. So if you're trying to do something quantitative where you're figuring out how many reads came from, say, the maternal copy of the genome versus the paternal copy of the genome, this is a common thing people want to do with RNA sequencing reads, for example, then this reference bias is perfectly confounded with the thing that you're trying to study. So for example, let's say we have a gene like gene 1 here. There's a maternal copy and a paternal copy of that gene. The paternal copy has 2 polymorphic sites, but both have the reference allele. And the maternal copy has 2 polymorphic sites, but only one of them is the reference allele. The other is some alt allele. Okay. So in this case, there's going to be some bias where it's going to be a little bit harder to place those reads correctly to the maternal copy of the gene, the one that has the alt allele. Maybe we're okay, right? Because read aligners are forgiving, right? They do approximate matching, so we can be forgiving of mismatches and gaps. And so maybe we're not so challenged in this case, but just think of another gene where you have an even denser cluster of alt alleles on, in this example, the paternal copy. What if the aligner can't deal with 3 non-reference alleles clustered close together? It, for example, blows away all the seeds that it might want to use in its seed and extend heuristic or something like that. And therefore, it might be very biased, in this case, against the paternal allele. And that's confounded with what we're trying to study, because allele-specific expression is trying to figure out, are there more transcripts from one allele or the other? But in this case, our ability to find those transcripts is reduced for one of the 2 alleles. That's one example. Another example, and this is actually probably in some sense, the prime example, because the very first paper that I know of that used the term reference bias for this kind of bias was an allele-specific expression paper from back more than 10 years ago now, this Degner et al. Paper from 2009. Another example would be hypervariable regions like the MHC region, which contains the HLA genes. These are important immunity-related genes. This region is hypervariable in the sense that if you compare 2 unrelated people, they probably have drastically different sequence in this particular region of the genome. And therefore, the reference genome is just, in some sense, it's just one choice for what the alleles might be in that region. If you go take reads from another individual and try to align them to the reference, you might find, as you see in this IGV pileup, you might see troughs in the coverage. Presumably, these are places where the alt alleles just clustered together densely enough that it just truly defeated the aligner, at least in those portions. Here's another example that I've seen firsthand that's a particular nightmare. So this is from the world of primary DNA methylation analysis. So this is bisulfite sequencing data. And there are 2 experiments. Well, there's one experiment, several experiments here where bisulfite sequencing data was obtained from several different mice. The mice were one of 2 inbred strains, black 6 or cast. And depending on which reference genome was used for the alignment, the black 6 reference genome or the cast reference genome, you would detect a differentially methylated region, but this was entirely spurious. There was actually no differential methylation here. The differences were due to genetic differences. So places where one strain had a CpG and the other strain had a TpG, which can be confused for having no methylation at a CpG. And depending on which strain you used as your reference genome, the direction of the differential methylation flipped, right? So if you use one strain, it goes one way, you use the other strain, it goes the other way. Complete nightmare, right? So it's another example of where reference bias is confounded with the thing that we're trying to study. Okay, and reference bias is not just something that bioinformaticians fret about. It's even been discussed in the popular press in some venues, since it's a big problem. First, what we really don't want is for example, our diagnostics and therapeutics that we develop using genomic technology to be differentially effective depending on what population we happen to have inside the reference genome that we were using. Okay, so this motivates the problem of trying to address reference bias, but there's good news, which is we can now turn to solutions. And it turns out that computer scientists and bioinformaticians have really been on this case for a long time. Again, going back a very long way. I can remember one of the very first conferences I went to, which was a genome informatics at Cold Spring Harbor. Dr. Schneeberger, the first author of that first paper there, was presenting Genome Mapper, which was a graph aligner based approach for aligning reads to Arabidopsis genomes. So there have been solutions going back a long way. People in the audience here will see their names here because they have participated in several of these breakthroughs. And they have generally had a couple of different flavors, and I'm going to touch on both those flavors today. One of them, which I will touch on first, is the idea of taking many reference genomes and sort of aligning and collapsing them together into a graph representation. So I'll spend a lot of time talking about graphs first. Another idea is to put collections of reference genomes into an index together without first trying to collapse them into a graph, and just letting the reference actually be a collection of genomes. I'll talk about both those kinds of approaches today. Okay, so let me start with graph-based approaches. And graph-based approaches have been proposed specifically to address this reference bias problem. So these quotes, which are from papers related to the VG aligner, are stating the reason why we want to put genetic variation into this kind of graph representation is in order to address exactly this kind of problem that we were just talking about, the reference bias problem. So in this picture taken from the VG paper, you can see the idea, right? So what we have here is a graph representation of a genome. There's going to be some portions of the genome that are common to all of the individuals who have been studied. Those can be collapsed into a single straightforward sort of node or path here. Then there's going to be places where there's variation. We have sort of forks in the road where we can choose one allele or the other. And then, another flat, more forks in the road. You get the idea. The variation is being encoded in a sort of graph shape where you can imagine yourself walking along the graph and sometimes having choices about whether to use this allele or that allele, right? As opposed to there being a special blessed allele that we call the reference allele, right? We're sort of letting there be equal choices about which allele might come next. So graphs can contain lots and lots and lots of variation, right? You could put in all, for example, of the variants from the entire 1,000 Genomes project into a graph. That would be a pretty complete graph, right? That would be a graph that in some sense encodes much of what we know. But the first question I sort of want to tackle here is the question of what should go in the graph. So let's make a distinction that will help us answer that question, which is let's distinguish between two different goals of why we might want to put variants into a graph. One goal might be when you put variants in a graph, you're really helping yourself to understand the relationships between those variants, right? If you let the variants be separate on sort of separate haplotypes and separate strings, you can't then easily reason about which might be, for example, the same variant, sort of at the same coordinate with respect to the reference genome. So when we collapse things together, it certainly helps us use this graph genome as a kind of catalog where we can understand not just what variants are there, but what's their relationship to each other? You know, which are the alternatives of each other, which are near each other? It helps with all such things. But another similar but slightly different goal that we might have is to use the genome graph as a map, right? Using it as a way of trying to figure out where reads go, you know, where a substring should be located. You know, a sequencing read, for example, should be placed with respect to the pan genome. In that case, more variation could be helpful because after all, every variant that we add is taking some information that we have about polymorphism in the population and putting it in the graph so that we don't have to penalize reads that have those alleles, right? We're sort of taking away penalties, which is good. On the other hand, we could wonder, could taking away penalties also be bad, right? Could taking away penalties eventually come back to bite us, for example, because it's making the representation too ambiguous? So this is the first question that I think we should ask, and it's going to help us to remember that there's these two goals, right? We can put everything in the graph, and then we have a nice catalog, or maybe we want to be selective if we're going to use the graph as a kind of map. So let me show a result from a paper from 2018 called Forge, where we looked at whether it would make sense to take genetic variants before putting them in the graph and sort of prioritize them and only take some of them and put some of them in the graph, but not necessarily all of them. And so what I'll show here is a result from a simulation experiment. Here, the horizontal axis, as we go to the right, we're including more and more variation in the graph, right? So the leftmost point is going to be a linear reference genome. The rightmost point is going to be a graph that has basically everything from the 1,000 Genomes Project. And then the vertical axis is a measure of what fraction of the alignments are correct, what fraction of the read alignments are correct. And because it's a simulation experiment, we can assess the correctness of the alignments that come out. And so you can imagine what might this picture look like in here, right? So for example, it could be that as we add more and more variation, the picture only gets better, right? And we only get more and more alignments correct. Alternatively, you could imagine maybe it's gonna get better and then get worse, where we get a benefit from adding variants at first, but then once we start to add too many variants, we start to get a drop. And in fact, I'll just show you the picture. You can concentrate only on this red line for today's purposes. It's really the only relevant part of the plot for today. And you can see, indeed, the red line has this property where as we add the first several variants, the highest priority variants, we actually improve our ability to find the correct alignments pretty quickly, right? This line goes up and it goes up pretty quick. Then we reach a point here, which corresponds, by the way, to around 8% of the variants from the 1,000 Genomes Project call set, at which point adding more variants starts to hurt, right? Adding more variants is actually hurting our ability to find the correct alignment, right? So this effect, the effect where the variants are helping, is because we're taking away penalties associated with alleles that really do exist and that therefore are in the individuals that we're simulating. The downward slope after a while comes because in the competition between how good it is to take away a penalty versus how bad it is to make the genome more ambiguous, eventually that second effect starts to win, right? So the ambiguity of the genome actually makes the aligner more confused. It starts to have more equally good choices for where to put reads, and now it's not functioning as a map very well anymore. Another remarkable thing I'll point out here is that when we do get peak performance, so this is sort of like the best we were able to achieve in this case with a graph genome. By the way, this is using the HiSat2 graph aligner. We actually make it most of the way to this point up here, and that point up there corresponds to the accuracy that we get when we align to a reference genome that is personalized. In other words, it has exactly the alleles that are present in the simulated individual. No more, no less. You know, it has only the alleles from that individual. So part of the message here is that the graph does quite well, but the graph does quite well when we are selective about what to include in the graph. And if we are too aggressive about including variation in the graph, we do worse. Something similar happens if you look specifically at reference bias. So if you take all the sites that are heterozygous in the simulated individual and ask what fraction of the reads that align to that site have the ref versus the alt allele. If there is reference bias, you'll see a tendency toward the ref allele, which is what you do see for the first several box plots here, right? They're off They're not centered on 50-50. They're a little bit above because there's a reference bias. But once we get to the point where we've included 10% of the variance in the reference graph, that's where finally we end up with a box plot that's centered on 50-50, and it stays there as we add more variance. Okay. So what did we learn from this set of experiments? We learned that if we are selective about what variance we can include in the reference genome, in the reference graph, we can effectively balance the pros and the cons, right? The pro of removing a penalty and the con of making the representation more ambiguous. We also learned that even a relatively modest number of variants, like 8 or 10% of all of those in the 1,000 Genomes Project call set, that even a modest number of those variants is able to alleviate bias and improve alignment accuracy all the way up close to the point that is achievable if we use a kind of perfect, ideal, personalized reference genome, which obviously is not really practical in reality, but it's a good sort of upper bound on what we can expect to achieve with a graph representation. Okay, and the peak accuracy that I showed you, it was at around 8 to 10% of the variance, 10% in the case of the reference bias experiment. That corresponds to, if we were to prioritize them by allele frequency, that corresponds to variants that are 5% or greater allele frequency, roughly. Okay, and this has been sort of built on and confirmed by others. This is another study that's quite similar. It uses VG instead of HiSat2, and it looks at cow breeds instead of human. Finds basically the same thing, that you need to sort of be selective in what variants to include in the reference genome, and that you can approach personalized accuracy. And since then, there's also been other work by, for example, Chirag Jain and others on other approaches for how to prioritize variants for inclusion in graphs other than the ones that we discussed in the Forge paper in 2018. Okay, so it was very interesting to us how small the percentage of variance was that we actually needed to include before getting peak performance. And it actually motivated us to try one other thing, a relatively simple approach, where here's what we did. So we thought, what if we basically take 2 passes to this approach of aligning reads to a reference genome? Let me emphasize right now, we are not going to use a graph genome in this framework. Graph genomes could fit into this framework, but we're not going to use them here. We're going to stay simple and use only linear reference genomes. And what we're going to do is start by aligning all the reads to a standard linear reference genome like GRCH38, and then ask, did the aligner seem to have trouble, right? Was there something hard about the decision that the aligner had to make to put the read where it went? And really all we do here is we pick out the alignments that have the reads that either fail to align or have low mapping quality, right? Basically the aligner thinks it may very well have got the alignment wrong when the mapping quality is low. So we pick them and we separate them by whether they seem to be difficult to align. And for the ones that were difficult to align, we subject them to another round of alignment. And we don't just align them to, say, 1 more genome, but we align them to 5 more genomes. And each of these genomes represents an individual from a different 1,000 genomes superpopulation. So these 3-character abbreviations you see here are the standard 3-character abbreviations that the 1,000 Genomes Project used for different superpopulations. So it's European, African, East Asian, South Asian, American admixed. And so for the reads that were hard to align or failed to align, they get forwarded to this kind of second pass. They get aligned to each of these population-specific reference genomes, all of which are linear. And then we take all of those results, the initial results for the reads that were easy to align, the second pass results for the reads that were hard to align, and we just merge them, right? So we say for each read, if we aligned it once, then take that alignment. If we aligned it 6 times, then we have to now pick the best alignment of all the ones we found, take that, and then possibly even have to change it a little bit, right? Because after all, the coordinate system is not necessarily the same. There are indels and other indels and structural variants that might be different between these different linear reference genomes. So we need to do a kind of liftover to get them back into the target coordinate system of GRCH38. And we just take the best and lift over for each read. Okay, you might wonder what fraction of the reads tend to make it to this second pass. And obviously this is going to vary, maybe even drastically, data set by data set, and based on things like how long the reads are and how high quality there are. But as a very rough estimate, in experiments we've done, it tends to be around 20%, around a fifth of the reads have to be forwarded to that second pass. Okay, so you might be a little worried about the fact that we seem to be doing a lot of work here, right? Instead of aligning the reads to one reference genome, we're aligning some of the reads to 6 reference genomes. But as I just argued, it's not all of the reads, it's sort of a fraction. It's maybe 1/5 of the reads that need to get aligned to all 6 of those genomes. And furthermore, you can do it in such a way that the peak memory footprint doesn't have to grow beyond the memory footprint of just one of these linear reference genomes, right? In other words, you can do the references in serial, sort of in the outer loop, if you will. And then you never have to have more than one of them in memory at the same time. Okay, and you can use, by the way, since these are linear reference genomes, right? Each of these is just a typical linear FASTA file that we have built somehow from the 1,000 genomes variants. You can use whatever linear aligner you like. We happen to use Bowtie 2, but you could use another one. You could use a standard linear aligner in this framework. Let me just show you a couple of results, and I'll do it quick. Let me call your attention just to these two sort of groupings of box plots here. What these are showing you, again, on the vertical axis, like before, is a measure of what fraction of the alignments, what fraction of the reads have been correctly aligned. And I'm showing you this for 2 different approaches. One of them is the approach I just described. And then the other is, again, a personalized sort of ideal reference genome that has exactly the variation belonging to the particular person simulated from. So sort of, we presumably can't do better than that. And you can see they're quite close, right? So they're quite close on alignment accuracy. So reference flow achieves nearly the same % correct as personalized. Here's a measure of reference bias, sort of like the second plot I showed you before. Focus on these 2 sets of box plots. So higher is worse. Higher here is more biased toward the reference allele. The reference flow approaches here. And here's VG, doing somewhat better than us, but both of us doing very close to personalized, which, no surprise, is sort of centered on that 50-50 line, right? A personalized genome should not experience reference bias. Okay, so we were able to get quite close to the sort of alignment accuracy and reference bias characteristics of a graph aligner like VG. So what did we learn? We learned that aligning to multiple linear genomes, as long as they are selected in such a way as to sort of cover the genotype space fairly well, in this case, we just used the different superpopulations, can achieve similar accuracy and bias as a graph aligner, but with a linear aligner that's very fast and memory efficient. So we can usually do it at a fraction of the speed and memory footprint. And to do so is really just a matter of writing a wrapper that goes around the aligner and building the appropriate indexes. It does involve that final liftover step, which actually involves yet another piece of software called Leviosam, which I won't have time to talk about today, but that's an important step. But that is also a solvable problem, that final liftover step. So what's the downside of doing it this way? Well, we're only using 5 genomes in that second pass. That's really the downside, right? 5 genomes is not a lot. If you think about a variant that's out there at allele frequency 10%, that variant has a good chance of not being in any of the 6 reference genomes that we would use in this pipeline. We're just not really covering the full space of polymorphism very well with an approach that selects a small number of linear reference genomes. And this probably explains why we were still lagging VG on that measure of reference bias, the one I showed most recently, right? Our box plots were like a little bit further up than the VG box plots, probably because VG is able to do a better job of including lots of variation in that graph compared to us and our 6 linear reference genomes. So what this does is motivate a different space of solutions that I'm going to spend the next several minutes of the talk focusing on, where instead of using a small number of linear reference genomes, is there a way that we can use a large number of linear reference genomes? We can include many entire panels of human reference genomes in the process of trying to figure out where reads came from. Okay, Before I get there, I'll just sum up by saying, I'm going to kind of conclude talking about graph-shaped reference genomes for now. And a wonderful recent advance that's worth everybody's time, and I saw 2 of these 3 authors in the audience when I looked earlier today, is Wheeler graphs, which are a framework that makes it that do a good job of generalizing a lot of approaches that have been proposed so far for how to index graph-shaped reference genomes. So this is one of the areas of theory that I really feel like has been advancing very nicely in lockstep with the practice. And now that some of this theory is set and known, and that theory is continuing to move forward, I think that's going to propel good future advances on both on the graph side of things and on the side that I'm about to describe for indexing collections of linear reference genomes. Okay. All right. So in order to talk now about how is it that we can index large numbers of linear reference genomes, let's say hundreds, thousands, panels of linear reference genomes, I'm going to slow down a little bit and talk through a little bit of conceptual framework for these recent advances. And it means that I just have to basically talk about the FM index for a little while. I like talking about the FM index. I think it's fun. Some people in the audience maybe have seen this before. Some people maybe dread seeing the slides that are trying to explain the FM index to them. But I really don't have to go into very much depth to explain what is the recent advance in this area. So let me just spend a little time on this. So the Burrows-Wheeler transform, which is at the core of the FM index, which is in turn at the core of alignment tools like Bowtie 2, BWA, and things like that. It's basically a way of taking a text. This might be a genome. It might be a collection of genomes. It might be 2, 3 genomes. A way of taking a text and simply reordering the characters of that text in such a way that the internal structure, repetitive structure of that text sort of comes to the surface. And I'll show you in what sense it comes to the surface. But in short, the way the Burrows-Wheeler transform reorders the letters is by putting them in order according to the alphabetical order of what comes to the right in the original string. Right? So each character finds its new position according to the alphabetical order of of its neighbor to the right, of its sort of context to the right. And what this tends to do is that if you have similar substrings, if you have repetitive substrings inside your collection, like if this is 1 reference genome, then perhaps these are repetitive elements. Or if this is 10 reference genomes, then it's perhaps the conserved portions of those reference genomes. So if there is a repetition inside the text, then after the Burrows-Wheeler transform, the characters that are near those repetitions are going to come together. And what you're going to have is runs in the Burrows-Wheeler transform. For example, let's say this is our text here. It's got sort of 3 variations on the word rectangle in it. So if you think about the characters at the beginning of those words, they all have this similar context that come to the right, right? They all have ectangle or something like that coming to the right. So once we take all these, once we take these characters and put them in order according to their right context, the R's are going to come together in a run, right? They all had very similar right context, so they all come together in a run in the transform text. These runs are the key to understanding why it's possible to make pan-genome indexes for collections of strings, basically because the runs are cheap in a way, right? If we can live in the world of runs as opposed to the world of characters, then we can really get by for cheap. I'll give you more examples. Here, for example, here's another string, and I'm about to take its Burrows-Wheeler transform. It's clearly very repetitive, right? You learn a lot about what character you'll find based on its right context, right? So if I take that and apply the BWT to find the transformed version of that string, I get something that has nice long runs in it, right? So you see runs of repeated characters. Pretty nice long runs. The average run length goes up substantially after we apply the Burrows-Wheeler transform. If the string is somewhat less repetitive, then we get somewhat lower average run length, but we still get some average run length that's almost always higher than the run length in the original text because we've brought things together according to their right context. Now just imagine what's this going to look like for human genomes? Like if we took a bunch of human genomes, let's say we took a bunch of diploid human genomes, and simply threw all of them together into the BWT, maybe concatenating them first, then what are we going to get? We're going to get that the length, of course, of this representation is growing linearly, right? As we concatenate more genomes, our concatenated string is growing proportional to the number of genomes. But the number of runs in the Burrows-Wheeler transform version is not growing linearly. It's growing much slower than that. It's growing sublinearly, because with each new genome, the number of runs is going up only, vaguely speaking, is going up only proportionally to the amount of new sequence in the new genome, and not proportionally to the amount of sequence, period. Right. So for example, if you were to build a series of strings out of an increasing number of diploid genomes, their length increases as the numbers that you see in the N column, but the number of runs in the Burrows-Wheeler transform increases only with the numbers that you see in the R column. So if you would like to be able to build an index that makes it possible to solve all our favorite sort of querying and indexing and seed and extend type problems, but for collections of genomes, the name of the game is you'd like to do it in terms of R. You'd like your data structure to grow only in proportion to R, and you'd like to try never to have a data structure that grows in proportion to N, because that data structure will be overwhelmed eventually as you add more genomes. Whereas a data structure that grows with R won't get overwhelmed as quickly, right? You might even be able to put thousands or tens of thousands of genomes in there before it gets too big. So that's the idea of a lot of these approaches. And these ideas about how to index and query in space proportional to R, these also go back a long time. And if you go back and think of the original FM index work, right, the work that Bowtie and BWA are based on, that happened back in around the year 2000. There was more work by Veli McKeenan and others. I saw Veli in the audience earlier today, to extend this so that it can do a subset of the queries that people are interested in doing with the FM index, but in space that's proportional to R, right? Again, that was our wish. We would like for the data structure to grow with R instead of growing with N. So we got partway there with these advances from more than 15 years ago. More recently, there was finally a breakthrough that allows us to do all of the kinds of queries that we would like to be able to do with this index, including the so-called locate query, which is the query that lets us figure out when we have a pattern match, where was it? Exactly where in what genome did that pattern match occur? Right now, even that query is something that we can answer using a data structure that fits in memory proportional to R. So this was only a recent advance, 2018 or so. And so this is, besides Wheeler graphs, which I already mentioned, this is another wonderful recent theoretical advance that I think is really going to benefit pangenomics, right? Pangenomics, we have basically two ways of thinking about things. Things live in graphs, or things live as independent linear strings. Either way, there have been some recent theoretical breakthroughs that are going to make it easier and easier to make further advances in the field. So in other work that was presented in RECOMB a couple of years ago, we showed a group of us together, people you see here in my group together, we worked on answering the question of how to construct these indexes in an efficient way. That was a serious problem with the original work, because it wasn't clear how to construct them. But we showed how to construct them in an efficient way. And we showed that if you use this approach to build indexes of very repetitive collections of human genome sequences, you could eventually beat lots of other approaches that we're used to using. So this is a very busy diagram. The important part is over here. And basically what it's showing that is, as you add more and more copies of chromosome 19 from different individuals, we're just basically taking chromosome 19 from a bunch of different 1,000 genomes individuals, and then ask how big is the index that you would build for that collection with different approaches. You first of all find that Bowtie eventually breaks, right? So it can't go higher than 250 genomes, basically because its index is growing linearly with N. So it eventually, will peter out. Whereas various approaches that are able to build indexes in compressed space, right? These Seq-based approaches, as well as the R-index approach, they all are able to grow much more slowly, and that once you get to a very large number of genomes, this R-index approach, this new approach, is able to build the smallest index of all. It's also the case that you can do the locate queries efficiently, though it's kind of a competition until you get up to the very repetitive collections. In other words, over here, Bowtie wins, but as you get up to the very repetitive collections, Bowtie not only loses, but eventually stops being able to build the index at all. So it's competitive on the speed side as well. And then maybe most importantly, these indexes are something that are practical to build over many human genomes. So in this experiment, what we're measuring on the vertical axis is something like the index size, although this way, in this experiment, we measured it as the peak memory footprint at alignment time, at indexing time rather. And we built indexes of larger and larger groupings of human genomes, where we either took the human genomes from the 1000 Genomes Project call set, basically taking columns out of the VCF file from the 1000 Genomes Project call set, or we took long read assemblies and just added more long read assemblies to the index. The difference being that the 1000 Genomes Project genomes probably don't have, or definitely don't have, the full complement of structural variation. The Human Genome Project VCFs generally only allow indels up to some length. Longer indels or more complex structural variants tend not to be there in those VCF files. If you take long read assemblies, you're getting everything, structural variants and everything else, also sequencing errors and other things like that. So, you know, it's not all good stuff, but you're at least not censored by some class of structural variants. And you can see that as you add more genomes, the memory footprint is growing not linearly, but somewhat sublinearly, okay? True for the long read assemblies, true for the 1000 Genomes Project individuals both. Okay, so that's the main thing I wanted to say about this second kind of idea, where we let the linear genomes be linear. And I'm going to argue that that's also a good idea, and one that we should be keeping in mind alongside these approaches that use genome graphs, right? When you let linear genomes be linear, it's easy, for example, to allow the structural variation to be there, right? Once you start to require that the genomes be in a multiple alignment with each other, or in some sort of alignment with each other, you're challenged by each new structural variant. It's sort of challenging your attempt to put these genomes in the same coordinate system. So approaches that leave the genomes in their initial linear state, but that nonetheless are able to build indexes that are compressed down to the amount of distinct sequence in those genomes, is very appealing. And I hope that these two things will continue to sort of grow up together. There's actually a couple of examples later in the conference of approaches, both of which are built on this R-index infrastructure, the infrastructure I was just describing, where we build indexes that grow proportionally to the number of runs in the BWT. One of them is discussed by my student Omar tomorrow at RECOMB-Seq, so check that out. And the other one is discussed by Max Rossi on Tuesday in the main RECOMB conference, so please check that out too. The main thing I'll say is that these indexes, what both Max and Omar are to some degree going to describe, is how these indexes allow for queries that are sophisticated enough that we can do, for example, mem finding, and we can do other things that are like mem finding, like finding matching statistics. And in particular, what Omar is going to talk about is we can use them to find matching statistics in an online and very fast way, so that it's useful in the context of, for example, the read until method for ejecting uninteresting reads out of nanopores. Okay, I'm almost finished, almost ready to conclude. I think I may even be a little early, which is kind of a miracle. One of the things I want to take away from, I'd like you to take away from this, is that when we talk about pangenomes, the question of whether you would like the pangenome to be inclusive or not depends, right? Being more inclusive and having, for example, a more efficient way of allowing more variants to be represented and queried is certainly a big benefit if your goal is to understand the variants and their relationship to each other. If, on the other hand, your goal is to use the pangenome as a kind of map, for example, as an index for aligning sequencing reads, then there's good reason to be selective. There's good reason to look at those variants and say, wait a minute, some of these are going to help me, but some of these are going to hurt me. And really all of them are going to do some of both. I need to be selective in order to figure out what to include in my representation. And so it's good to keep the fact that there are two different applications of the pangenome in mind so that you can use those separate ways of reasoning in the appropriate places. Another takeaway, I hope, is that linear aligners, while using a single linear reference certainly has all the problems that I outlined early in the talk, linear aligners, if given just a bit more information, for example, a handful more linear reference genomes that are chosen so as to be genetically distinct and to cover a sort of genetic space, they're able to do very well and actually to be competitive with graph aligners on the measures that really matter to us, including reference bias. I talked finally about the R-index, and in particular, I think the R-index is going to be useful in a future where long-read human assemblies are not going to be like a once or twice a year thing anymore. It's going to be that they're coming fast and thick, and we're going to need to do something with them. And if we want to be able to use them, for example, as a catalog of all the structural variation that's available from long-read assemblies, it's going to be difficult to put them into common coordinate systems together first, right? To find the multiple alignment that relates them inclusive of all their structural variation, right? So approaches that let linear genomes be linear, let these various assemblies be linear, are a good set of tools to have in our toolbox going forward. Okay, and then finally, final conclusion slide that's a little bit more zoomed out. One of the things I think is particularly exciting and gratifying about pangenomics now is that the theory and the practice are really growing together. It's not like one is lagging far behind the other. And I think that's going to create more opportunities. And I hope that it keeps sort of both theorists and practitioners interested for a while longer. These formalisms like R-index and Wheeler graphs and other things I didn't have a chance to talk about today, but that are also very important, like, for example, minimizers, strobmers, things like that. We heard from Christopher Sahlin earlier today. You know, these are all very important ideas that mostly have a good theoretical underpinning, but that we still need to drive the theory forward before we really understand what they're doing and what we could be doing better. And then finally, if you're interested in learning more about these formalisms I spoke about, things like FM index, things like Wheeler graphs, a little bit about R index, or at least some of these ideas underlying R index. I have a playlist on YouTube that you can check out. So the link is there. Feel free to check it out. Okay, thank you. I want to thank all the people who contributed to the work, who are all here on this slide. Thank you very much to my funders. Don't forget that two of these folks, Omar and Max, Omar is speaking tomorrow in RECOMB-Seq. Max is speaking Tuesday in RECOM. Please check out those talks. Finally, I'm interested in hiring a postdoc to work in this area. So if you're interested, just send me an email and I'll tell you more about that position. Thanks, everyone. Happy to take questions.
Fabulous. Thanks so much, Ben. Well, it looks like we've got a set of questions already queued up in here. So this first question from Victor, I think, goes back to some of your early slides, and we'll see if we can figure out. He was asking, do colors represent populations? Oh yeah, I think that was on the VG, the graph from VG slide. I think in that example, the colors represented individual haplotypes, right? So the VG graph is able to encode that branching structure corresponding to what alleles are there. But furthermore, it's also able to encode what are the paths through that graph that correspond to particular individuals that were indexed. So I think what you were seeing with those, it's a little bit like a subway graph, right? It's a subway picture, right? You were seeing the particular colored lines corresponded to which alleles an individual had. Great, thank you. All right, our next question is from Camille. Excellent talk, Ben, thank you. I'm wondering if you have ideas on how work on the SR index fits into the future of pangenomics. Yeah, okay, so let me see. I might get this wrong, but I think the SR index is the one that samples somewhat differently depending on whether the
depending on whether the runs are long or short. I may have that right. If I don't have that right, then my answer won't be appropriate. But I think that that's the kind. I think that basically what that kind of method is helping to do is, if you assume one thing about how repetitive your collection of genomes, if you don't want to assume much about how repetitive the collection of strings is that you're putting into the indexing approach, then an approach like SR index would help you, because even if you're wrong and they're not very repetitive, it's still able to adjust the suffix array sample so as to not be so dense. The thing about the R index is it always wants to choose its samples to be at the beginning and ends of runs. So if the runs are not long, then the sample is actually quite dense. But the SR index, if it is the approach that I am thinking of, and I've got the name right, it basically helps you because you can have a sparser sample even where the runs are short, and you can basically pick the sampling. In some sense, you can pick the sampling scheme most appropriate to that stretch of the BWT.
All right, our next question is from an anonymous attendee. And so is there a difference between DNA reads and RNA read alignments?
Yeah, I mean, this whole talk has been chiefly about what happens if you apply these methods to alignment of DNA sequencing reads. In a sense, it all applies to RNA sequencing reads too, but yes and no. Because finding alignments for RNA sequencing reads, as we heard, like, for example, this morning in the Ultra talk, right, it often involves a seed finding step, right? So that seed finding step is a pattern matching problem. And all of the indexes we discussed are indexes that could service that pattern matching problem. But the frameworks that I discussed, like the reference flow framework where we align to many genomes, I would say that's a little trickier, because part of that framework is trying to understand, it's hard to assess, first of all, because it's hard to say what's a correct alignment. Is it just an alignment that got in roughly the right spot? Or is it an alignment that got in the right spot and had exactly the right splice junctions in all instances? The definition of what's correct starts to get slightly muddied in the RNA-seq space, and that certainly affects the measurement. It probably also affects our decisions about what we think is a suspect alignment. An alignment where, for example, it seems very convincingly to have a junction, but the junction splice motifs don't match what we expect. That might be a good example of something we would want to forward to the second pass. So I think the shorter answer to your question is, in theory, especially because a subproblem here is seed finding, yes, all these approaches apply to DNA and RNA reads. In practice, I think everything that I discussed would need some special adaptation and care if it was going to be applied to RNA sequencing reads.
And our next question is from Martin. Do long reads also suffer from reference biases?
Yeah, I mean, like, yes and no, right? Yes, yes, in the sense that all those same phenomena happen, right? There's the long reads also have alt alleles, right? So in that sense, it's the same story. But in another sense, imagining the reference flow framework, for example, long reads tend to be able to figure out where they go, right? Even despite the fact that they have many mismatches and gaps, just their sheer length usually gives you plenty of good information about where they go, which means that if your threshold for whether something deserves more attention in a second pass is that it has a low mapping quality, probably only a small proportion of reads will really have a low mapping quality for long reads. All the points still apply. It's just that it's probably a lower proportion of reads that are getting forwarded to that second pass. It might also mean that the magnitude of the biases, like the ones I was discussing early on, is perhaps not as high, because you're more able to get the correct alignments for reads when they're long. I don't think that there's yet a really clear picture of how much we suffer from reference bias from long reads, or at least not that I know of. Someone will probably send me a reference. But you also generally get lower coverage with longer reads that can exacerbate things. You also obviously have sequencing error issues with longer reads that can also exacerbate things. So on balance, what it probably does is reduce the number of reads that need special care and attention. But I would assume that all the same problems are there, perhaps just at a lower they're not perhaps not bothering us as much, but they're all certainly still there, I would guess.
All right, our next question's from Iman. Great talk, Ben. Yeah. Can you please briefly talk about how the last liftover step of your rep flow method handles structural variants?
Great question. Yeah, it doesn't. But you know, everything that we do in the reference flow framework is depending on these 1,000 genomes call sets, which, like I was saying, they're not, you know, they're basically structural variant free, right? They're only allowing insertions and deletions up to some length. And therefore, the liftover problem, we don't need to worry in those cases about structural variants. However, my student Nate Chun, who did that great work on Leviosam, his current focus is exactly on this question of when we are dealing with real reference genomes that are from long read assemblies, say, where the best information we have about the relationships between these genomes are coming not from a multiple alignment, but from, say, a pairwise alignment that yielded a chain file or something like this. How do we do liftover as best we can? And obviously with structural variants, the answer will never be simple because reads can span, you know, reads can partly span a structural variant, reads can span part of an inversion or something like this. And therefore, there's not really a clear single destination for the read. Reads sort of perhaps turn into chimeras after liftover, or perhaps reads can't be lifted over at all because they are placed entirely within a private insertion in a reference genome. So the work we've done that I spoke about on reference flow can't consider these cases. It just limits itself to data sets that are described with VCF files that are described with existing multiple alignments. But in the future, we think some of these ideas, well, we think pretty much all of these ideas can be adapted. It's just that we will sometimes have to live with partial answers on the other end of the liftover.
Great. Next question's from Ryan. Fantastic talk, Ben. 2 questions. I keep getting confused by those 2 ways to include variation in a reference, either modify the graph or linear reference and change the bases directly, or add new nodes to the graph. Did you talk about both ways or just one or a mix of both? And let's answer question 1 and then I'll read question 2.
Sure, yeah. In the case of graph aligners, I was always talking about essentially adding more nodes, adding more complexity to the graph, changing the shape of the graph to accommodate those variants. So for example, in the Forge work, we were giving HiSet to more forks in the road. You know, we were creating more elaborations to the graph. That was also true for I mentioned that cow work that used VG. Also true in their work. They were giving it more variation. It's also the case in that work that when we built personalized reference genomes that had exactly the alleles from the individuals, that we were representing heterozygous variants the same way with forks in the road in the graph, right? Bubbles, maybe you can call them. So in all of that work, it was always about elaborating the graph. In the case of the reference flow framework, everything was linear. Right, so the genomes were all selected so that they always had a particular unambiguous chain of alleles, but the individuals were chosen so as to be kind of spread out in genotype space, or at least we trusted that by picking individuals from different 1,000 Genomes Project superpopulations, we were getting individuals who were kind of spread out. So in the case of the reference flow work, and in the case of all the work that I talked about subsequently with R-index, that's all about taking collections of linear genomes and indexing those. And so it's not about creating new elaborations in a graph, it's just about including more linear sequences in the index.
Great, and I think this is going to be the last question that we'll answer live, and I'll ask Ben to answer any of the other ones that have come in via text. So regarding your conclusion, why couldn't graph aligners avoid bias by also adding multiple references, same as linear aligners?
They definitely could. And in fact, in the reference flow work, where we say in the discussion section, there's no reason why these have to be linear genomes. We chose linear genomes because we wanted to demonstrate the power of even typical linear aligners to overcome reference bias if you give them a little bit of a hand. But there's no reason why you couldn't, like, for example, you could change the whole approach so that at each of those spots where you're using an aligner, you're using VG. And you could even change it so that instead of there being pass 1 and pass 2, there could be a whole chain of passes. And you could be going, for example, from a more inclusive graph to progressively less inclusive graphs. Or perhaps the other way. Sort of depends what your goal is. But you could imagine if you're only forwarding along the reads along this flow where there was ambiguity in their alignment, then you can imagine there's an advantage to starting with your most inclusive graph and then going to less inclusive graphs as you go along. So the framework absolutely allows that. You can, in a sense, you can plug in whatever aligner you like in those different boxes. We focused on a linear aligner because we wanted to make the point that, similar to the Forge paper, that you don't have to walk too far into the space of genetic variation before you can do a great deal to address reference bias.
Great, thank you so much. I think with that, we've hit our time. So thanks to everyone for a great session this morning, depending on where you are in the world, or afternoon. So we're going to have a